Welcome to Section 4 of Molecular Biology. In this section, we'll be discussing translation. Let's get started. This is an overview figure of transcription and translation in both eukaryotes and prokaryotes, which can be found in Section 3 of Molecular Biology. In the last video, we discussed transcription. Recall that this is a process that uses DNA as a template to synthesize mRNA. In this video, we'll be discussing translation, which you can see right here. Translation is a process whereby polypeptides are synthesized using mRNA as a template. So mRNA is converted into a polypeptide. As we go through translation, we'll show it from the perspective of a eukaryotic organism, but the process is similar in both eukaryotes and prokaryotes. This is a detailed figure of translation which can be found in Section 4 of Molecular Biology. On the right side of the figure, we can see tRNA right here. On the left, we can see the small ribosomal subunit and the large ribosomal subunit, as well as mRNA. Eventually, the two sides of the image converge to form a ribosome, mRNA, and tRNA complex that is responsible for synthesizing a polypeptide. You can see that right here. Okay, let's go through each step of the figure, starting with tRNA. Transfer ribonucleic acid, or tRNA, is a molecule that transfers amino acids to the growing polypeptide. The molecule contains four important regions that you need to be familiar with. The D arm, which you can see right here, facilitates the binding of tRNA to the enzyme aminoacyl tRNA synthetase. The anticodon loop, right here, contains the anticodon that pairs with the codon in mRNA. From the figure, you can see the mRNA codon, AUG, right here. In this case, the anticodon of this tRNA would be UAC. So if we erase this little blue circle, we can see that better. AUG pairs with AUC. We'll talk about this step more in a second. Sometimes the anticodon in a tRNA molecule is still able to pair with an mRNA codon, even if the bases don't perfectly complement one another. This is known as wobble pairing. For example, if we assume the mRNA codon is CUU, the anticodon in tRNA could be GAC. In other words, the first two bases of the anticodon pair perfectly with the mRNA, which you can see right here, but the last base, uracil, does not pair perfectly. Normally, the uracil and mRNA should pair with adenine, not cytosine. Despite this, the tRNA molecule would still contain the appropriate amino acid, which would be incorporated into the polypeptide. So again, this process right here is known as wobble pairing. The T arm, which you can see right here, facilitates binding of the tRNA molecule to the ribosome. Finally, and most importantly, the acceptor site right here is the region that's responsible for binding to an amino acid. For step 1, you should not only know that this is the acceptor site, but also know that the 3' end, which you can see right here, contains the base sequence CCA. CCA are the only bases shown in the figure. However, if you were to look at all of the bases on the tRNA, you would see a large number of modified bases such as pseudouridine, dihydrouridine, and ribothymidine. These modified bases are important for formation of the tRNA secondary structure, which we can see looks kind of like a T. Okay, moving on. Notice that aminoacyl tRNA synthetase right here is the enzyme responsible for combining a molecule of tRNA with an amino acid. You can see the amino acid is now bound to the tRNA right here. Also notice that ATP is required for this to occur. The addition of an amino acid to tRNA is also known as charging. You should also know that aminoacyl tRNA synthetase has the ability to scrutinize or proofread the amino acid. If the amino acid is incorrectly bound to tRNA, it can hydrolyze the region between the amino acid and the tRNA. In the figure, this reaction is shown as if it's repeated 3 times. The 3 tRNA molecules are shown right here. And notice that each contain a different anticodon and corresponding amino acid. Okay, now let's focus on the left side of the image and discuss ribosomes. Notice that the strand of mRNA right here finds the small ribosomal subunit right here. The large subunit is initially separated from the small subunit, but will later combine with the smaller subunit. In the figure, you can see that the large subunit has 3 sites: the A site, the P site, and the E site. The A site is responsible for accepting charged tRNA molecules, so A for accept. The P site processes the tRNA or facilitates the formation of a polypeptide, so P for process. And finally, the E site is where tRNA molecules exit the ribosome, so E for exit. Okay, before we move on, you should know that ribosomes are different in eukaryotes and prokaryotes. For example, the large subunit is comprised of the 40S and 60S subunits in eukaryotes, whereas in prokaryotes, it's comprised of the 30S and 50S subunits. The small ribosomal subunit is also different. However, rather than memorizing these subunits, just know that for now, these differences allow antibiotics to target prokaryotic ribosomes while sparing eukaryotic ribosomes. There are 3 steps to polypeptide synthesis, and they include initiation, elongation, and termination. From the figure, you can see that initiation occurs when initiation factors and GTP are used to bring the large and small subunits together along with mRNA and tRNA. Okay, before we move on to elongation, you should know that there is something called the Kozak sequence. This is a region around the start codon that facilitates the binding of the small subunit to the mRNA. So it would be this region right here on the mRNA. The exact sequence isn't as important as knowing that mutations to this region can impair the initiation of translation. You should also know that the start codon is usually AUG. You can see that right here. Some students remember this with the phrase AUG inaugurates protein synthesis. Rarely, the start codon can be GUG. Elongation occurs as the tRNA and mRNA molecules move through the ribosome, resulting in a growing polypeptide. Notice that the amino acid serine, right here, was added to methionine. You can see that right here. And notice that now the tRNA in the ribosomal P site contains a growing polypeptide chain. Also notice that as this process continues, the ribosome moves along the mRNA towards the right side, like this. This means the mRNA is read in the 5' to 3' direction, just like replication and transcription. Finally, you should know that specific elongation factors are required for this step to occur. A great example of this is elongation factor 2, or EF2. This is an elongation factor which is inhibited by exotoxins produced from Pseudomonas and diphtheriae. Termination occurs when a stop codon in the mRNA is encountered, which you can see right here. The stop codons include UAG, UAA, and UGA. Some students remember these by the mnemonics "you go away," "you are away," and "you are gone." Notice that releasing factors, which you can see right here, bind to the ribosome at the stop codon site, and that the entire complex then dissociates, resulting in a finished polypeptide. Also, pay special attention here. Notice that the stop codon does not code for an amino acid. From the figure, you can see that the releasing factor is occupying the A site that corresponds to the stop codon. Therefore, the codon just prior to the stop codon will code for the last amino acid in a protein. After termination, the polypeptide can undergo post-translational modification. This just means other molecules can be added to the polypeptide. Examples of this would be methylation, acetylation, or others. Okay, let's solidify this information with some questions. Researchers are studying the process of translation in mice. In one particular mouse model, a knockout mutation results in embryonic demise. Upon further analysis, the researchers discover that the mutation results in modified bases inserted at the 3' end of molecules normally responsible for binding amino acids. They also notice that in this particular mouse model, the activity of aminoacyl tRNA synthetase is decreased. What is the most likely normal composition of the 3' end of the mutated molecule? From the question stem, we're told that the mutation results in modified bases inserted at the 3' end of a molecule normally responsible for binding amino acids. This should make you think of tRNA. Let's pull up the overview figure so you can see what the question is referring to. From the figure, you can see a molecule of tRNA right here. Notice that the 3' end contains the base sequence CCA right here. In the question stem, it alluded to the idea that this 3' end was mutated. As a result, the activity of aminoacyl tRNA synthetase was decreased. So again, the normal composition of the 3' end is CCA. Okay, let's do another question. A 7-year-old unimmunized boy presents with a sore throat that started 3 days ago. He has also noticed malaise and low-grade fevers. On exam, he has cervical lymphadenopathy, pharyngeal erythema, and coalescing patches of gray fibrotic tissue directly adjacent to his tonsils. The physician suspects that this patient's symptoms are due to an infectious organism that produces an exotoxin. This exotoxin most likely inhibits what factor associated with eukaryotic translation? Hopefully from the question stem you notice that this boy has signs and symptoms consistent with Corynebacterium diphtheriae. The combination of pharyngitis, so sore throat, with a pseudomembrane, which is indicated by the coalescing patches of gray fibrotic tissue, is classic for this infection. Corynebacterium diphtheriae produces an exotoxin known as the diphtheria toxin, which inhibits elongation factor 2, or EF2. You can see that elongation occurs right here, so this step would be inhibited by the exotoxin.